Hello, everybody. In this lecture, we're going to talk about nucleic acid structure, which should be a little bit of a review. We're going to go a little bit more in depth than we did before. So let's get right into it. By the end of this lecture, you should be able to describe the structural features of DNA and RNA nucleotides, relate directionality of nucleotides to the formation of these phosphodiester bonds, describe the structural features of RNA and how they differ from DNA, and explain the flow of genetic material as it is presented by the central dogma. So, when we start out, nucleic acids are pretty much what govern all of cell life. It is going to determine your cell structure and all of its functional capabilities. Now, we've talked about this a little bit, but nucleic acids are made of three parts. We have a phosphate, a sugar, and a nitrogenous base. Now, in DNA, that sugar is deoxyribose. In RNA, that sugar is ribose. The major difference is on this number 2 sugar here, which we call the 2' carbon. There is no OH in deoxyribose, therefore deoxy, that's without oxygen, while ribose has an OH there. The phosphate, when it's in its form before it's been added to the DNA strand, this is a triphosphate, so we would call these DNTPs, or basically DNA nucleotide triphosphates. So that tri meaning 3 phosphates. Once it is added to the DNA strand, it's going to cleave off those 2 phosphates, which we call a pyrophosphate, and that's going to provide the energy necessary for polymerizing your DNA. And it just has 1 phosphate in your backbone. Now, notice here we can have one of 4 nitrogenous bases in DNA: adenine, guanine, cytosine, or thymine. And we can have one of 4 in RNA, and in that case, thymine is subbed out for uracil. What's important here also is that we have numbering of our carbons in our sugar. And so if we go from our oxygen, go clockwise, our 1' is where our nitrogenous base is added. The 2' is going to determine whether it's deoxyribose or ribose. The 3' is going to always have an OH, regardless if it's deoxyribose or ribose. The 4' is going to have the 5' attached to it, and the 5' has the phosphate. And so these bases that are on that 1' carbon are going to be divided between purines and pyrimidines. Your purines are adenine and guanine, while your pyrimidines are cytosine, thymine, and uracil. Now, when we bind our 2 strands together, we're going to get this double-stranded molecule that curves around, and it resembles kind of like a twisted ladder or a spiral staircase. And we call this a double helix. Now, this is going to be largely determined by base pair bonding. They make up the rungs of our ladder, and they're going to be bonded complementary. So A binds to T and G binds to C. When A binds to T, it has 2 hydrogen bonds, and when G binds to C, it has 3 hydrogen bonds. So it's a slightly stronger bond when you have GC than it is when you have AT. Now, if this were RNA, you can still get base pair bonding, but instead of A going to T, A goes to U, still complementary. Now, these side rails are going to have this alternation of sugar phosphate. This is what we call our sugar phosphate backbone, or our phosphodiester bonds. And so to reiterate what I've already said, the phosphate is on your 5' carbon, the OH is on your 3' carbon, and the base is on your 1' carbon. And it's always going to be set up in this format of being built 5' to 3'. So if we were building this strand of DNA, this would be our first nucleotide because it's at the 5' end. The 3' OH is going to bind to this new NTP at the 5' phosphate group. So you get a diester bond or a phosphodiester bond. You get an ester bond here and an ester bond here with a phosphate in the middle. That's what gives us phosphodiester, which we can see here. Now, making bonds takes energy. And so when this phosphate gets added on to this OH, we need to provide energy to do that. And the way that happens is that this phosphate is usually a triphosphate, and then it'll cleave off the pyrophosphate, which is the two terminal phosphates of your triphosphate. And that's going to catalyze or give the energy necessary to bind to this OH. Giving you this sugar phosphate backbone where you have a 5' phosphate and a 3' OH that repeats. And so we can see that kind of here, where we start out with our 5 to 3, we make this bond. This becomes our phosphodiester bond. So this is one ester bond here, one ester bond here, phosphate in the middle. Now, on a linear piece of DNA, you're going to have one end, which is your 5' end, that's always going to have a phosphate. And then one end, which is your 3' end, which is always going to have a hydroxyl or an OH. Now, if this is a circular DNA like we have in prokaryotes, that 5' and 3' are going to be bound together. And so this directionality is really important because when we build our DNA, it's always going to be built 5 to 3. This is going to result in a couple of interesting characteristics of DNA. So if you imagine that you have a paperclip, one end of it is your 5' end and one is 3'. This is represented by yellow and purple. When we connect two paper clips together end to end, it's going to go 5 to 3. So it's going to go yellow to purple, yellow to purple. The yellow end binds to the purple end. If we go even further and make more pieces, you're going to just get a longer chain where you get a 5 to 3, 5 to 3, 5 to 3, so forth and so on until you get to the very end. So one end is 5 and one end is 3. Now, if we have two different chains of paper clips, then they're going to be organized in what is called an anti-parallel fashion, where one end is going to go 5 to 3 from top to bottom, and the other end is going to go 5 to 3 from bottom to top. And so this is what is going to lead to our what is known as anti-parallel setup of DNA, where one side goes 5 to 3, and then the 5' end of the other strand is going to bind to the 3' end of the first strand, and it's going to go 5 to 3 in the opposite direction. So they're parallel in the sense that they are going side by side, but they're anti-parallel because that directionality is different. So 5 to 3 from top to bottom, 5 to 3 from bottom to top. And that anti-parallel structure is what allows for these nucleic acid bases, these nitrogenous bases, to actually bind together properly. Now, RNA is very similar to DNA. There are a couple of differences, which we'll kind of highlight here. One similarity is that they're still made up of your three basic parts, which is a phosphate, the ribose sugar, and a nitrogenous base. The big difference is that uracil is subbed in for thymine. The ribose sugar is going to have this OH on the 2' carbon instead of just a hydrogen, which is what deoxyribose has. You're still going to have your base on the 1', and you're still going to have this 5 to 3 relationship where we have a 5' carbon up here bound to a 3' OH, and then you're going to get this kind of ester bond, phosphate ester bond, making a phosphodiester bond, and it's going to go generally in this 5 to 3 direction. Now, we think of RNA as being single-stranded, which is true, but it can still have secondary structure by folding over on itself. And so RNA doesn't exist by having a double strand like DNA does, but it does maintain some stability by binding to itself. And so these bases want to be bound to something else. They don't do well by themselves, so they fold over. So we get all these different structures. And I'm not interested in you memorizing how these different structures are formed. What I do want you to think about is the fact that these structures do exist at all. Now we have three primary RNAs that we're going to talk about in class. Messenger, transfer, and ribosomal, which are mRNA, tRNA, and rRNA. We'll talk more about those as we go through this chapter. But it does allow us to think about the central dogma of molecular biology, where DNA is transcribed to RNA, and RNA is translated to protein. Now, sometimes RNA can be reverse transcribed to DNA, but our central dogma is DNA is transcribed to RNA, which is translated to protein. And that's what these future lectures are going to be more about. So with that, you should be able to describe the structural feature of DNA and RNA nucleotides, relate directionality to those phosphodiester bonds, describe how RNA is structured and how it differs from DNA, and explain that central dogma of DNA to RNA to protein. With that, I hope you enjoyed, and I'll see you soon.